At the Institute for Protein Design, I'm a translational investigator leading development of our protein nanoparticle platform for therapeutic applications. I came to the University of Washington because during my PhD I began reading David Baker's work and realized that Rosetta is really the leading tool for designing and engineering proteins with new structures and functions. So this model is the very first self-assembling protein that we ever designed. This is an octahedral assembly of 24 subunits, so it kind of looks like a cube, and there's a protein trimer on each corner of the cube. So this has octahedral point group symmetry. We used a pre-existing trimeric protein as the building block to design this structure, and what we did is we docked these trimeric protein building blocks symmetrically in this configuration and then designed a new protein-protein interface between the trimers that drives assembly of this material and stabilizes it. So this is an example of what we call a two-component assembly, where you have a gray protein and a blue protein that co-assemble to this architecture. You can pull out two building blocks, one dimeric blue protein building block and one pentameric gray protein building block, and then you can also break those apart, and you can see in green the designed interface highlighted. So this was the portion of the protein surface where we used the Rosetta software to introduce mutations that would stabilize this particular geometry between these two protein building blocks, which when repeated symmetrically leads to this icosahedral nanomaterial. To get closed or bounded structures like cages or spheres or anything like that, you essentially have three choices in terms of your symmetry group. You want to use a cubic point group symmetry, and those are tetrahedral, octahedral, and icosahedral symmetry. Of the three, icosahedral symmetry tends to give you the largest internal volume for packaging things, and also a larger number of subunits, which for multivalent display for vaccines is the smallest one that we've designed is a one-component tetrahedron, 12 subunits, that's about 11 nanometers in diameter. And the largest we've designed is a two-component icosahedron, 120 subunits, and it's, I think, 35 or 36 nanometers in diameter. So this is, I hope I don't break it, is Young's one-component icosahedron I301 in blue, so that's a 60 subunit homomeric icosahedral assembly. And in green, we can see 120 copies of GFP green fluorescent protein that have been scaffolded on this nanoparticle through genetic fusion. So this is a very highly fluorescent designed protein nanoparticle. I think the main applications that we're targeting really are vaccines or targeted delivery. So you can see a company pursuing one or both of those with these designed protein nanoparticles. First and foremost, we have to thank David Baker, who has made all of this possible through the development of the Rosetta software in collaboration with the Rosetta community, which is really a vibrant worldwide community now. And also for putting together the Institute for Protein Design, which is kind of taking all of this work to the next level. We've been very blessed to have a long list of extremely talented collaborators. So the Yates group at UCLA has done a lot of crystallography in support of this work. Tamir Gonen's group at Genelia has done really just incredible electron microscopy on this. We have lots of collaborators in the vaccine space. We're signing some up in the targeted delivery space. People here in the lab like Young, Jacob Bale has really driven this recently. Will Scheffler has come up with the creative and brilliant computational solutions that really gave birth to these efforts. Additionally, the people that have funded this work, the Gates Foundation has been a huge supporter of our nanoparticle platform, and that's been really enabling. DARPA has supported this platform. Takeda Pharmaceutical Company, God, the list goes on and on. That is just ridiculous. And then it breaks down into monomers with like scaffolded GFPs. That's so funny. Holy God. Those guys really put a lot of effort into this. Wait. Okay.